Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic enzymes. This is from the chapter biomolecules. So watch the entire video lecture to gain basics out of it. So without any delay, let's start with the topic enzymes. Let's start with the topic enzymes. So at first, what are enzymes? Enzymes are biological catalyst which influences the speed of biochemical reactions. Alright. So they are catalyst which enhances the speed of the any biochemical reactions. Now all enzymes are protein, but all proteins are not enzymes. So you have to know that all enzymes are proteinaceous in nature, but all proteins are not enzyme. And the enzymes are specific. Alright. Enzymes are specific, and each enzyme has its own substrate. So again I repeat, enzymes are specific and each enzyme has its own substrate. Ribosome is also an enzyme like a nucleic acid RNA that behave like enzyme. So what is ribosome? Ribozyme, not ribosome, it is ribozyme. Alright, so you have to understand this is not ribosome, this is ribozyme. So ribozyme, wherever so you can find in nucleic acid, that is in RNA, they behave like enzymes. So enzymes form tertiary structure, that is 3D, with some services or pockets called active site into which the substrate fits. Alright, so enzymes form the structure, tertiary structure. You can see the lock and key hypothesis. Enzyme is a functional shape and you can see the substrate. Alright, so enzymes form a tertiary structure 3D with some pockets or cervix, and these are called active site in which substrate binds. Next, coming to chemical reactions. So chemical compounds undergo two types of changes. One is physical change and a chemical change. Alright, so chemical compounds undergo two types of changes. One is physical change and a chemical change. So physical change. So what is a physical change? A change in shape or state of matter without breaking bonds. Alright, so a change in shape or state without breaking bonds. Example, ice melts into water and water becomes vapor. Alright, so ice melts into water and water becomes vapor. There is a physical state. So ice is converted into water and water into vapor. Now coming to chemical state, what is chemical reaction? In this, the bonds are broken and new bonds are formed. Alright, so in chemical change, the bonds are broken and new bonds are formed, and it may be organic or organic. Alright, it may be organic or inorganic in reaction. Example, we have inorganic and organic. For inorganic, you can see barium hydroxide plus sulfuric acid. Alright, so barium hydroxide plus sulfuric acid gives barium sulfate plus water. So again, for organic one, you can see starch plus water gives glucose. Alright, so we can see the structures given, the rate of a physical or chemical process, that is the amount of product formed per unit time. Right, so what is rate? The amount of product formed per unit time. So chemical compounds undergo two types of changes we have studied. One is a physical change and the chemical change. All right, now a rate is called velocity if direction is specified. So the rate in which the direction is specified is called velocity, and the rate of physical and chemical processes are influenced by factors such as temperature. Alright, generally rate doubles or decreases by half for every 10-degree change in either direction. So rate of enzyme catalyzed reaction is very high. Example, carbonic anhydrase is the fastest enzyme. So carbonic anhydrase is the fastest enzyme. It accelerates the following reaction, alright, 10 million times. So carbon dioxide plus water, it gives to H2CO3, that is carbonic acid, on the presence of enzyme carbonic anhydrase. Alright, so carbon dioxide plus water gives H2CO3 in presence of enzyme carbonic anhydrase. So in the absence of this enzyme, only 200 molecules of carbonic acid are formed in an hour. Alright, so in presence of carbonic anhydrase, about 6 lakh molecules are formed per second. All right. So in absence of enzyme, 200 molecules are formed, whereas in presence of enzyme, 6 lakh molecules are formed per second. So you can see the difference. All right. So these are the functions of enzyme. It means it enhances the rate of reaction. Now, in a metabolic pathway, each step is catalyzed by different enzymes. Example, in glycolysis, 10 different enzymes takes part. Alright. So glucose is converted into pyruvic acid. So in glycolysis, you can see 10 different enzymes takes part. Hexokinase are there, phosphofructokinase are there, phosphoglucoisomerase are there, aldolase are there, pyruvate kinase are there. Many enzymes are there. Next, we'll see the nature of enzyme action. The catalytic cycle, right? Catalytic cycle, we'll see. So enzyme acts with substrate like a lock and key model action. All right, like one key will open this one lock. All right, so it includes the following steps, like substrate binds to the active site of an enzyme, so E plus S. All right, next is this induces some changes in enzyme so that the substrate is tightly bound with active site of the enzyme to form enzyme substrate complex. So you can see when enzyme plus substrate binds, it forms an enzyme substrate complex. You can see in figure below given E plus S forms ES. Alright. Next is the active site breaks chemical bonds of your substrate to form enzyme product complex. Alright. Now the enzyme substrate breaks and forms a different product that is called enzyme product complex. And the last step is the enzyme releases the product and the free enzyme is ready to bind to the other molecules of the substrate. Alright, that is E plus P. So you can see an enzyme, you can see a product. So this pathway goes through some unstable transition state structure. Alright, so enzyme plus substrate gives enzyme substrate, then it forms enzymes product, and then it gets separated into a different enzyme and product. Now we'll see the concept of activation energy. How do enzymes speed up chemical reaction? All right, how do enzymes speed up a chemical reaction? We'll see. So you see the concept of activation of energy or activation energy. You can see on left hand side the chart is given, but one side is the y-axis is the potential energy and x-axis is the progress of reaction. So you can see substrate, if substrate is higher, the product is higher, right? If the more the substrate, the product is higher. You can see the activation energy without enzyme and activation energy with enzyme. So you will see that activation energy is the additional energy required to start a chemical reaction. All right, so activation energy is the additional energy required to start a chemical reaction. So in order to start any reaction, activation energy is required. So in an exothermic reaction or endothermic reaction, exothermic means heat is evolved, endothermic means heat is absorbed. Alright, so in an exo or endothermic reaction, the substrate must grow. Alright, the substrate must go through a much higher state, alright, to a much higher energy state. This is called transition state energy. Therefore, activation energy is different between average energy of substrate and transition state energy. So again I repeat, therefore activation energy is the difference between average energy of substrate and a transition state of energy. So if the product P is at a low energy level than the substrate, the reaction is an exothermic reaction or spontaneous reaction, and if it requires no energy by heating to form the product. So in a biochemical reaction, the enzyme lower than activation energy. As a result, the speed of the reaction increases. Alright. Now, what are the factors affecting enzyme activity? You can see if temperature and pH, concentration of substrate, and presence of inhibitor. Alright. So you can see if I increase enzyme, the temperature also increases. As we increase enzyme, the temperature also increases as the enzyme. Right. So temperature is increased, enzyme activity is increased at a certain point, and again it drops. Same way for pH. As we move in a higher pH, enzyme activity increases and again falls. And next for substrate concentration, first it increases, move into saturated point. Now you can see temperature and pH. You can see enzyme shows highest activity at optimum temperature and pH. All right, so enzymes show highest activity at optimum temperature and pH, and activity declines below and above the optimum value. So you can see the curve, that is the inverted U-shaped curve. That means the activity declines at below or above optimum value. Alright, so at low temperature the enzyme temporarily is inactive. So at low temperature the enzymes becomes inactive, whereas at high temperature enzyme destroy. So again I repeat, at low temperature enzyme becomes temporarily inactive, whereas at high temperature the enzyme get destroys. Alright, the enzyme get destroyed because proteins get denatured by heat. So this is all about temperature and pH. These are highly effective. So temperature and pH, alright, so highest activity at optimum temperature and pH. So at lower temperature it's become inactive, at higher temperature it get destroyed by denaturation of heat. Now inorganic catalyst work at high temperature and pressure, but enzyme get damaged at high temperature above 40 degrees Celsius. So thermophilic organisms have enzymes which are stable at high temperature up to 80 to 90 degrees Celsius. So thermophilic bacteria can work at a temperature higher than 40 degrees Celsius, that is up to 80 to 90 degrees Celsius. Alright. Now moving to concentration of substrate. So with the increase in substrate concentration, the velocity of enzyme action rises at first and reaches a maximum velocity, that is Vmax. Alright. So whenever maximum velocity is reached, it's called as Vmax. So this is not exceeded. Alright. So this is not exceeded by further rise in concentration because enzyme molecules are fewer than the substrate molecules. So no free enzymes molecule to bind with additional substrate molecule. So they are low substrate, they are high substrate. Okay. So you can see the Vmax, there's a maximum velocity. Now presence of inhibitor, how presence of inhibitor affects the factor of enzyme activity. So the binding of specific chemicals like inhibitors shuts off the enzyme's activity. This is called inhibition, right? So whenever there is a presence of inhibitor, it shuts off the enzyme's activity, and the inhibitor closely similar to the substrate is called competitive inhibitor. So the inhibitor closely similar to a substrate is called competitive inhibitor. It competes with the substrate for the binding site of an enzyme. So as a result, the substrate cannot bind and the enzyme action declines. So you can see below non-competitive inhibition and competitive inhibition. For non-competitive inhibition, you can see one enzyme is there, one substrate is there, one inhibitor is there. That means substrate will bind, substance are unable to bind with the enzyme. It's in the active site, but the inhibitor is binding at the opposite side. Whereas in the competitive inhibitor, we have seen enzymes are there, substrate are the inhibitor are there, where substrate and inhibitor competes to bind with a specific site. Whereas in non-competitive inhibition, the substrate and inhibitor don't compete for binding up. Next, moving to the examples for competitive inhibition. So you can see malonate is similar to the substrate succinate, alright. So malonate is similar to the substrate succinate. So it inhibits succinic dehydrogenase in the following reactions. Succinate, in presence of enzyme succinic dehydrogenase, forms fumarate, alright. So here competitive inhibitors are used to control bacterial pathogens, alright. So competitive inhibitors are used to control bacterial pathogen. Now I move to the classification and nomenclature of enzyme. Classification means how we classify, and nomenclature means how we name the enzyme. So there are following types. You can see six types are there. One is oxidoreductase, or it's also called dehydrogenases. Next is transferase, next is hydrolysis, next is lysis, next is isomerase, and last is ligases. So we'll see this one by one. First is classification and nomenclature of enzyme. First is oxidoreductase oblique dehydrogenase. Here it catalyzes oxidoreduction, that is between two substrate, right? So one is a reduced substance, another is oxidized substance, and forms, you can see, oxidation and reduction takes place. So in this process of oxidoreduction, dehydrogenase, here oxidation and reaction takes place. Next is transferase. Transferase means it will transfer a group, like catalyze a group transfer other than hydrogen. All right, so it will transfer from one group to another. All right, you can see AB plus C, it later forms A plus B and C, or B gets moved. Next is hydrolysis. Hydrolysis means what? It catalyzes hydrolysis of ester, either peptide, glycosidic, carbon-carbon chain, C halide, or PN bonds. All right. So you can see AB plus water, it gives to AOH and BH. Next, moving to lysis. So what is lysis? The catalyzed removal of group by mechanics, all right, other than hydrolysis, leaving double bonds. So isomerases are there to catalyze interconversion of opticals, geometric or positional isomer. And ligases, it catalyzes the linking of two compounds together. Example, we have enzymes which catalyze the joining of bonds like CO bond, then carbon and sulfur bond, then carbon and nitrogen, then phosphor and oxygen bond. Now there are certain cofactors are also there. So you can see cofactor plus apoenzymes forms holoenzyme. So apoenzyme is in an inactive state and holoenzyme is an active state. So you can see these are non-protein constituent bound to an enzyme to make the enzyme catalytically active. So apoenzyme, protein portion of an enzyme. So apoenzyme are the protein portion of the enzyme. So you can say apoenzyme plus cofactor gives holoenzyme. So when the cofactor is removed from the enzyme, its catalytic activity is lost. Alright, so whenever any cofactor is removed from an enzyme, its catalytic activity is lost. Now we move to different types of cofactors. So there are three types of cofactor. One is prosthetic group, coenzyme, and metal ions. Now what is a prosthetic group? So organic, that is tightly bound to apoenzyme. Alright, so the any substance that is tightly bound to apoenzyme are called prosthetic group. Example, heme in peroxidase and catalase that catalyze breakdown of hydrogen peroxide to water and oxygen. So peroxidase and catalase. Now what is a coenzyme? So organic, like transient binding to apoenzyme. Alright, so many coenzyme contains vitamins. Example, NAD, NADP contains niacin. Okay, so example of coenzyme is NAD. So these coenzymes are organic in nature. Now next, third is metal ions. What are metal ions? They form coordination bond with side chain at active site and one or more coordination bond with a substrate. Example, zinc is a cofactor of carboxypeptidase. So zinc is a cofactor of carboxypeptidase. Thank you, student. This was all about the entire video lecture. We have discussed about the topic enzymes. Hope you have liked this video lecture and have understood. So this was all about the topic enzymes from the chapter biomolecules. So stay at home, stay safe. I'm Shomuji sir signing off for today, and again we'll meet in the next class. Thank you, students.